Hello and welcome to this next video on OCR A-Level Biology. We're continuing with Module 2.2 and this is Biomolecules. And in this video we are going to be focusing on lipids. So let's get started. So there are three major types of lipids we need to know for the exam. So the first one, let me just write it here. So this first one is triglyceride. Now, before I go into this, lipids are also organic molecules, but they contain large amounts of carbon and hydrogen, and very minute amounts of oxygen. So they are non-polar, which means they are insoluble in water. But they don't dissolve in water, but they will dissolve in alcohol. So these are macromolecules, but because that's not the same molecule joined into another one of the same molecule over and over again, these are not polymers. So lipids are not polymers. So these are triglycerides, and this here is a triglyceride. And triglycerides are formed from this molecule, which is shown here, and this molecule is called glycerol. Basically, it's an alcohol. It's propane 1,2,3 triol. That's its proper name, but its biological name is glycerol. And this here is a fatty acid. It's actually made up of 3 fatty acids. And fatty acids are these long hydrocarbon chains with a carboxyl group with a COOH group attached. Now when a glycerol and a fatty acid join together, they eliminate water. So water is eliminated in the process by what we now know as condensation. And they form a bond here, which is called an ester bond. So you can see that there is an ester bond. And if you want to break apart, you need to add water in, break apart this ester bond. Do you just get the glycerol and fatty acid again? You need to do hydrolysis. And that requires water in order to break this ester bond to form glycerol and the fatty acid. Now this here is an example of a monoglyceride, because it's glycerol with one fatty acid. However, if we do this step 3 more times, then we get something called a triglyceride. So this structure here is a triglyceride. Now just a bit about fatty acids. So fatty acids come in many different sizes, and they can be either saturated or unsaturated. What do I mean by that? Well, saturated means that there's no CC double bonds. So there's none of these CC double bonds. And if it's unsaturated, that means there is at least one CC bond, CC double bond. So we use the same thing for like alkanes and alkenes. So alkanes, we say in chemistry, alkanes we say are saturated because there's no double bonds between carbons. Unsaturated, that's alkenes, and they have at least one CC double bond. So having one or more of these CC double bonds changes the shape of the hydrocarbon chain, because these bonds restrict rotation. That means that it introduces a sort of a kink in the molecule, and because these kinks push the molecules apart slightly, it makes it more fluid. And this is very important when we get to biological membranes. later on in the series. Later on. So that was all triglycerides. But there's another type of lipid, and it's down here. So this is a phospholipid. And phospholipids are similar to triglycerides, that they have a glycerol, 2 fatty acid tails, but instead of like 3 fatty acids attached to glycerol, it's only 2. And that is because on the glycerol there is a phosphate group attached, where the third fatty acid should be. So we call this a phosphate head of the phospholipid. Hence it's called a phospholipid because it contains a phosphate group. So this is the phosphate head, and these are the 2 fatty acid tails. And because of this phosphate head, phospholipids can have some unique properties when they're in water, and some unique behavior when they're in water, because of their unique property in that they're amphipathic. Whatever that word is, I can't say, and I'll write it down here. So phospholipid molecules are this. So it's this word which I can't say. Let me try it. Amphipathic. 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 That's what I was trying to say. So whatever that word is, that word I can't say. That's what these phospholipids have. And the reason for that is because this phosphate head is hydrophilic. And because it's hydrophilic, it means it's water loving. So it loves to be around water. Whereas the fatty acids, as you already know, these are hydrophobic, so they are water repelling. So hydrophilic means literally water loving. Hydrophobic means phobia is like a fear, so they are hydrophobic, so they have a fear of water, so they want to be away from water. And because of that, these phospholipids have a very unique behavior in water. So if I draw these phosphate heads, you see when you put phospholipids in water, they'll either form one of 2 structures. One structure we don't need to know about too much, but you just need to be aware. The other structure, which I'm drawing, you do need to be aware of. So this structure here, which I'm drawing, with the phosphate heads outside pointing towards water. Remember, they're hydrophilic, so they're water loving. It is weird, they love water. And then the fatty acid tails, they're hydrophobic, they have a fear of water. So this kind of structure is called, let me just finish these fatty acid tails. So this structure is called a phospholipid bilayer. So this is a phospholipid bilayer. I can't spell today, can I? Oh dear. Phospholipid bilayer. So what that means is these phosphate heads are outside on the surface of the bilayer. So these are all on the surface, which means they're around water. And these fatty acid tails are trying to get away from water, so they point inwards. So remember, for a phospholipid bilayer, you need the phosphate heads on the top and the bottom, like on the outside, and the fatty acid tails need to be on the inside. So this is a phospholipid bilayer, and it's really important for forming plasma membranes. So plasma membranes, the cell surface membrane, nuclear membrane, mitochondrial membrane. What do we have? Chloroplast membrane, Golgi membrane, endoplasmic reticulum. All of those organelles that I just mentioned are made from this plasma membrane, which all contain this phospholipid bilayer. The other structure, which I won't go into too much detail, because you just need to be aware of this. This one you actually need to know about. The other one you just need to be aware of and that's called a micelle. Micelle, it's either micelle or micelle. I don't know. Micelle. And basically, there's a picture on page 63 showing you what a micelle looks like. But yeah, that's pretty much all on phospholipids. No, I do not want to do that. Oh, I've just realised that we've only done 11 minutes for my recording here. Okay, that's fine. Anyway, let's move on to cholesterol. So cholesterol is the final structure that we're looking at today. And this cholesterol is basically a sterol. Cholesterol, which is you can see down here, this is a sterol, which literally means it's a steroid alcohol. It consists of 4 carbon-based rings, you can see here. And these carbon-based rings, you don't really need to know like the technical terms, unless you're doing like biochemistry at university or something. This is actually really, because I'm currently doing a biochemistry module in my uni so far, so oh my god, I can't type and speak at the same time apparently, because I'm doing a biochemistry module as part of my degree, so yeah, this might be really helpful for me as well. But anyway, cholesterol, this is a steroid alcohol. It's made up of these 4 carbon-based rings, which we call isoprene units. You don't need to know this technical term, you just need to know it. That's just for extra information. It's a hydrophobic molecule, so again, it doesn't like water. And it's mainly used in like cell surface membranes. Where it's used in cell surface membranes to help regulate the fluidity and maintain stability of the membrane. And more on that in module 2.5, which is all about which is a whole module on, which is a whole topic on the cell surface membrane itself. It's made in the liver. Oh, for plants, I mean, I don't know much about plants because I'm not a plant person. No, as in I'm not doing any degree. My degree is not related to plants, it's more related to like animals, specifically humans. Plants, they have a cholesterol derivative, and that's called, it's a really weird word, stigmasterol. That's how you pronounce it, I think. But yeah, and it's only different with only one respect. It has a double bond between carbon 22 and carbon 23. That's all. And another thing about cholesterol is used to make steroid hormones. So the steroid hormones that some of you should be aware of, you may be aware of, are testosterone, I can't type, estrogen. Estrogen is also made from cholesterol, so estrogen. What are the hormones are steroid? Oh, vitamin D. Vitamin D is made from cholesterol, I think. Yeah, vitamin D is made from cholesterol, even though it's not technically a hormone. These are all steroid hormones, which means they are all derived from cholesterol. So this is the molecule of cholesterol down here. And yeah, I finished this in record time. Anyway, that's it for this video. In the next video, we're going to be continuing on module topic 2.2, which is biomolecules. And in the next video, we're going to be looking at proteins. And there's a lot about proteins you need to know. So after that video, we'll move on to inorganic ions, which will be a short video. And after that, we're moving on to like practical biochemistry. So thank you for watching. Hope you enjoyed it and found it useful at the same time. And I'll see you in the next video. Bye.